beautiful beach behind me. In today's module, we're going to be focusing on some of the examples of the application of decomposition ecology. The learning outcomes of this fourth module on decomposition is for you to demonstrate an understanding of how decomposition ecology can be applied to real-life problems. For the first example, we're going to briefly leave this nice beach behind us. One important application of decomposition starts with the problems arising from the introduction of European cattle into Australia in the 1880s. In addition to the clearing of land for grazing, cattle caused another problem. And that problem comes in the form of the waste cattle leave behind, like this cow dung here. If you've ever had the pleasure to examine cow poo, you'll recognise that it is generally moist and soft. This is in stark contrast to the Australian native herbivores, such as the kangaroo, that produce small, hard, dry and fibrous dung. If you've ever had the pleasure to examine cow poo, you'll recognise that it is generally moist and soft. This is in stark contrast to the poo of Australian native herbivores, such as the kangaroo, that produce hard, small, dry, fibrous dung like the one you can see here in this picture. The problem with the soft poo of cows is that the larger native Australian detritivores, such as beetles, have not evolved to use cow dung, and as such, the decomposition process is very slow, with cow dung pads remaining on pasture land for months and even years. The consequences here are twofold. Firstly, given the amount of waste a cow can produce, it reduces the area suitable for grazing. Secondly, the dung becomes a breeding ground for flies and parasitic worms and other pests. With approximately 2,000 flies being produced from one cow pad and each cow producing approximately 12 pads per day, you can see how the population explosion of bush flies quickly becomes a serious public health issue. In fact, up until the 1950s, bush flies was such an issue that outdoor eating was prohibited unless the area was exclusively enclosed by fly screens. The solution was to look at places where large herds of native cattle exist, such as in Africa. In these habitats, detritivores, like the African dung beetle, have evolved alongside the cattle and their poop, and so are part of the natural ecological solution to organic waste removal. If you're not familiar with the dung beetle, these industrious insects do the job of physically removing the waste cattle leave behind. They either mix it into the soil, or the most famous examples you'll find are those dung beetles that produce large dung balls like the one you can see here in the photo. You might be asking, why would they do such a thing? Well, it's part of their breeding cycle. These beetles use the dung to create nutritious nests for their offspring. For those beetles that create these dung balls, a female will dig a hole into the ball, lay a single egg inside before sealing it up again. The larvae that then hatch have a ready-made meal available to them. The introduction of the dung beetle from Africa began as a CSIRO project in the 1960s and continues today to be one of the biological control success stories for Australia. Over 25 species of dung beetles have been introduced and established, resulting in a huge reduction in the numbers of flies. For each new introduction, care is taken to consider the ecological consequences, and the beetles are produced in specialised facilities before they're being released. If you're interested in knowing more about the history of the dung beetle in Australia, I recommend you take a look at this YouTube video here. It's a little bit dated, but it's got some excellent videos of these dung beetles doing their job. Our second example involves some detective work. Decomposers are very useful for the reconstruction of historical events. This is because they are abundant, can be found pretty much everywhere, and we know that certain material can decompose in a relatively predictable fashion. Therefore, decomposers have a long history of use in forensic sciences, specifically the field of forensic entomology, that looks at the succession of species arrival through egg laying, larval stages, and maturity to determine things like time of death and location of death. Two methods are generally used to estimate time since death, including the successional waves of insects from blowflies through beetles and other flies, These are useful if an individual has been dead longer than a month before discovery. And maggot age and development, which is often more useful for the time of death if the time of death occurred less than a month before discovery. Therefore, it is important that forensic scientists are careful not only with the collection of samples for investigation, but that they take accurate and detailed notes on other factors, including data on the time of day, location of the body, and specific site of the body where samples were taken, moisture content, temperature, and the state of decomposition. Forensic entomology is successful because of a lot of previous work on the biology of decomposers and their life cycle. This means we know that the time between each life stage remains fairly constant at a given temperature. We can also use size of each larval stage to determine the death accurately to several hours, and pupa casts can be used to estimate the time of death for older bodies. Sounds to be reasonably straightforward, right? Well, that was before ecologists started to look more carefully at what influences decomposition and found that variability in decomposer communities and their succession can impact forensic data. Just a quick warning, the next images shows a decomposing carcass and might be a little off-putting for some of you. We can see that the variability of succession of decomposers in the photograph here, showing replicate decomposing swine carcasses. Only 2 replicates shown in panels B and C have large fly masses and would be suggested of different time of death from the other 4 individuals. What do you think might be influencing this variability? We now know that insect arrival to carrion can vary based on factors such as insect population genetics and phenotype, the abiotic conditions, the biotic environment, such as the presence of competing organisms, as well as several unknown factors at play. All of these aspects point to a need to strengthen forensic scientists to gain more reliable data. Most forensic scientists focuses on the post-colonization stages of a decomposer behavior. That is, the time after the detritivore has consumed the decaying matter, and relatively little attention has been paid to the earlier pre-colonizing stages, including their initial exposure to the carrion, the detection, their final acceptance of the decaying matter. These aspects are important because forensic entomologists are now learning to appreciate the impact of variability at these early stages and what that has in the estimation of time of death, pointing to a need for a new framework that incorporates both ecological and evolutionary aspects into forensics. There's been much debate over quantitative measures of decomposition, and these measures are often still inaccurate. For example, prior to the 1990s, decomposition was measured in discrete stages based on calendar days, known as the degree days of decomposition index. Now, forensic researchers are considering the uses of under-indices, such as the accumulative degree days, that models incorporates environmental variables such as average day temperatures. As biologists, we learn to accept that the natural world has variability, but for forensic investigations, that variability can lead to errors in conclusions, such as time of location and death. These are fundamentally important to the investigation. So it is important that forensic scientists too have a working knowledge of the processes of decomposition and the ecology and evolutionary forces that are affecting variability in that process. This paper here nicely details some of the arguments and opportunities for increasing the focus of ecology and forensic science. The use of models, such as swine carcasses that we saw earlier, have been traditionally used for forensic studies, but they can have their limitations. And there is an increasing number of human decomposition facilities that are available, otherwise referred to as body farms. This includes one in Australia, which is the Australian Facility for Taxonomic Experimental Research, here in Sydney. Other technological advances are improving our understanding of decomposition and the ecology, and these include, for example, DNA sequencing technology that's being used to characterize microbial decomposers at very fine scales and over large timeframes. Okay, For our third example of applied decomposition ecology, we've just moved from a pristine beach into an environment that most of us would prefer to ignore. Human waste is a massive ecological problem. In Australia alone, we toss out about 20% of the food we buy, and food waste comprises about 40% of the average household waste. So, decomposition ecology is relevant to you. Based on what you know about the process of decomposition, pause the video for a moment and take the time to think about what happens to food waste that ends up in landfill like the one behind me. Yes, waste starts to decompose, but once it is buried under all that rubbish, conditions become anaerobic, and that's when the processes of fermentation and methanogenesis come into play. Through these microbial processes, decomposition of food waste into landfill sites results in the release of potent greenhouse gases, including methane, which is about 25% more potent than CO2. But there is a solution to preventing food waste from getting to landfill in the first place, and that is through composting your food waste. You can do this via a worm farm or compost. The key is to ensure you keep your system aerobic by mixing it regularly to prevent methane from being produced. Check out this review in the Journal of Global Change Ecology if you want to know more about emissions and waste management. You can also find some helpful tips and advice from your local states and council websites, where you can find where to compost if you don't have access to it in your own system. Now, let's head back to the beach. That's all I have for now. I hope you've enjoyed these modules on decomposition. Next time you find the last week's sandwich in the bottom of your bag looking a bit like this, take a time to not just screw up your nose, but to stop and think about the complexity and importance of the decomposition process you're witnessing. And rather than toss it into the regular bin, compost it. See you all next time.